So hello, and thank you very much for joining our webinar on drug development. For anyone who doesn't know me, I'm Zach Whiteley. I'm chair of the Acute Leukemia Advocates Network. Delighted to have you with us. The intention of this webinar is that it will be participative. So if you have any questions, if you're on Facebook, then please comment them on the Facebook live stream. And if you're here on Zoom, then please comment them in the normal way using either the Q&A or the chat function. I'm delighted to be joined by Samantha Neer, who is the network manager of the Acute Leukemia Advocates Network, who's going to give us an overview of how the drug development process works, all the way from preclinical through to clinical. So over to you, Samantha. If anyone has any questions, please post them in the chat and the Q&A, and I'll be keeping an eye on those throughout.
Thanks, Zach, and so welcome everyone. So happy to be here today. So the intention today is really to go through the whole process of drug development. I guess some of you have already attended some webinars on clinical development, but actually the process is a bit longer than that and is a bit more complex than only clinical trials. So here on the slide, you can see the whole process, and basically it goes for a more than a 10-year plan. So the time that you need to develop a drug is over 10 years. And you actually go from about more than 10,000 compounds to one single drug approved, which means that there's only about 2% of substance evaluated that actually make it to the market. So it's not so much. And so if we see here the number of drugs in the pipeline, you can see the comparison between 2019 and 2020. And you can also see quite strangely with COVID, so we would tend to think that the research has kind of slowed down or stopped. But when you look at the numbers, it's actually quite interesting to see that the number of drugs is quite similar compared between the 2 years. And when you see the number of drugs in preclinical phase versus the number of drug launch, you can actually see the about 2% that I was mentioning just before. And again, that's more than a 10 years plan, and the cost is just huge. So develop a drug is a huge cost. So it's an average cost 400 million to 1.5 billion. And it's actually the cost before the medicine can actually be made available to patients. And from there, the reimbursement process starts. So it's a lengthy and very costly process, as you can see. And when we talk about drug development, so as I said before, it's not only about clinical trials, there's all the phases actually before the clinical trials. And that involves research and IP, so intellectual property, the regulatory landscape, the production and the quality, and then also as the preclinical and the clinical phases. And actually, all of these triggers the rationale to initiate or not the development of a drug. So, and when we talk here about the process, it's a general process. So we all know that there's exception to the process, and you may be already familiar with those exceptions. So we just here take the very, very general process on how drug is developed. And it's a process followed by the pharmaceutical industry. We also know that some researcher can come up also with some research and medicine, but let's say here that we go through the process that pharmaceutical industry has to follow. So the kind of question that they have to ask themselves before actually initiating the drug development is how does the drug work? So they have to know about the structure, the properties, the mechanism of action, all the biological activity, and then what kind of data are already available. So what's already published in the literature, what do we have some in-house data already? And a very important question for, especially for industries, who is owning the intellectual property? Because that again costs a lot and can really make industry to go or not to go for a drug. They also have to evaluate all about the disease and the unmet medical needs. So I'm not going to go through all the questions here, but they really have to know everything about the disease process. What's the unmet needs? Is the disease part of another disease family? Do we have some history about that drug or about the disease already? Is it a field already crowded in the space? And then what do we know also about the regulatory context? So are there endpoints that are already approved? Do we need to go in special markets? What do we do? What do we know already in the literature? What are the quality of life factors? So many, many, many questions that they have to ask themselves before actually initiating or not a drug development. And then obviously, as you may imagine, there's also all that has to do with the market. So they have to understand what's the market size and what the competitors landscape is looking. So really to know how many patients are in key markets. So how many new cases per year, what's the market growth, what's the market segmentation, how many patients are living with the disease. But also they have to start thinking about a sales forecast. So what would be the treatment model? So how many cycles of treatment, the duration, and then what would be the targeted price for the treatment per patient. Plus, they also need to actually position the product versus competitors. So again, a lot of questions that they have to ask themselves. You can see all the questions here, and you can read that. But so it's not a clear cut on yes, no. There's a lot of things to know before you actually start investigating in a drug. And there's also a bit of comparison to do. So this is the kind of things that they use. They would put the description of their candidates and then do the same thing for their competitors and future competitors. So then they would be able to position the drug with all the information they know. And that would kind of indicate already whether it's a good, sorry for the word, market opportunity or not. And then there's also to take into account all the parts related to production and quality. So do we have enough supplies for the R&D, the preclinical and the clinical development? What do we know in terms of quality and comparability? So everything which has to do with good laboratory practice, good manufacturing practice. What do we know about formulation and stability? Do we have stability studies already? What would be the analytical methods and the bioassays to support the R&D and preclinical and clinical? And then they also need to understand in terms of regulatory, what do they know? So at what point they would need scientific advice from the different authorities like the FDA or the EMA. Do we have some specific guidelines available already? What the ICH guidelines are saying? And then would it be possible, for example, to get an orphan drug status? What would be the strategy, the regulatory strategy in key markets? Because as you know, many the countries have different strategy for that. And then what would be the implications while running clinical trials? So yeah, so all of those questions have to be answered before starting the drug development. And so as mentioned before, you can see it's not a straightforward process. There's a lot of things to take into account. And it's a quite complicated decision to make for industry. And here's the development milestones. So as mentioned before, it takes about 12 years, and it goes various different steps. The first thing is to discover, so doing the research, and then you have to validate the research, so that then goes into the preclinical, and then you have to develop, so then the drug is going into the clinical phases. And when we talk about drug discovery, it is the process on how the new medication are discovered. Before, it was mainly by chance, or that the drugs were actually discovered. And then the more the diseases are getting complex, the more complex it gets as well to discover new drugs. So before, they also used to, they were using the classical pharmacology, and because there were existing chemical libraries, so you could compare the drugs with the already existing libraries. And then there was all about the human DNA sequence. So by reversing the pharmacology, then you could find new remedies. And today it's an extremely complicated process because there are a lot of pathways that researchers are discovering. The diseases are getting more and more complex because the genetics is getting also more complex. So all the process is very, very complex. And so the first thing you have to do when you do the research is to find a target. That means, what will the drug affect? Which means you have to find a drug for disease. So the researcher, they really have to understand the cause of the disease, the conditions, and then through that they can understand the process, how the disease works, what's the pathway. And then from there, they would say, okay, then we need to target that kind of mechanism. And that's actually the starting point. And the target, so that would be the target. That's actually the first point. And it's already a quite complicated process. It's already taking, as you can see, between 12 and 18 months to actually identify a target for a specific disease. And probably they would identify different targets just to make sure that they have different they have different solution afterwards. And once they have identified that target, then they need to identify the compounds which actually have an effect on the target. That's called the hit discovery. So what compound can actually hit the target? And the tricky thing here is to understand what compound can hit the target, but not interfere with other targets. So it's a very, again, complex process. It goes through some special screening that they call HTS. It's large libraries of data that they have already of chemicals tested. And then they would compare the target versus the hits. And usually they screen about more than 10,000 compounds in that process. And once you have all those targets, then you need to narrow down the selection of the hits to actually go to a lead. So that means that they would screen all those compounds, and then they would slowly start to narrow down the selection with different methods. So they will compare with some database. They would do some virtual screening. They would use some existing database, and just to see which compound is actually the more optimized for what you want to see. And that's also a long process. And then once you have this lead compound, it's really then to understand which one you can make even better. So they would it's very unlikely that you would find a perfect drug candidate right now, except if you have a lot of chance. So then they would start to modify a bit the structure of the molecules to see if they can try and improve their activity. And then again, it narrowed down the numbers of compounds that would actually be going afterwards into preclinical. So basically, all this process of drug discovery and research is really to narrow down the number of compounds, and you go from 10,000 potential compounds to 1. And then this is where the process actually stops. When you have 1 candidate, then you can move into preclinical. So here again is the process. You would be able to read that afterwards. It's and then you get your candidate that then you want to validate in preclinical. So the preclinical studies are in vitro studies and in vivo studies on animals to test the efficacy of the compound and also to be able to do some toxicology studies. So they would try to understand what would be the dose, let's say the best dose before starting to use that in humans. They would also try to understand what would be the relevant route of administration. They would also try to see if this has some effects in pregnant women and in women who could potentially have kids afterwards. There are also some studies that they can do for pediatric trials. And in this preclinical, they would also look at the pharmacokinetic and the pharmacodynamic. So it's really to understand how the body would react to the drug and how the drug would impact the body. So a lot of studies that they actually have to conduct to validate that the candidate could potentially go into to be tested in humans. And with those data, so with the preclinical data, then you are allowed to file for an IND application with the FDA. It's also a quite cumbersome process. There's a lot of information to be provided to the agency, which obviously makes sense because you need to know if your candidate is safe to be tested in humans. And this is then, once you get a positive feedback from the FDA, this is where you can actually start your clinical testing. And so there's probably most of you know already, there's different phases in clinical trials. And so you start with a low number of patients, and the more you advance, then the more patients you get in the trials and the longer the trials are running. The phase 1 trials is usually a very small population. So this again is a very general process, and we know there's some exception. So in general, there's less than 100 healthy volunteers. We also know that in the case of cancer or rare disease, the phase 1 trial can enroll already patients and not healthy volunteers. And the purpose of this trial is really to determine, is the treatment safe? So they would look at dose escalation. They would try to understand what's the highest dose that the body can take without any serious side effects, how the drug does to the human body and how the human body actually reacts to the drug, and also to understand if it's safe with other medication, and also to understand what's the best route of administration. So would that be an oral drug or an IV or topical? And placebos are usually not used in phase 1 trials. So placebos is inactive treatment. And the statistic shows that about 70% of the drugs moves from phase 1 to phase 2. The phase 2 trial is basically what's happening in the phase 1 trial, except that you have more people enrolled in the trial, and the duration of the study is longer. So then then you could see more side effects. And it's to understand how the treatment works. You would monitor your patient for several months to understand how effective is the study treatment and how is it safe for the patient. It's a bit premature to understand the overall safety of the medication, but the phase 2 trials actually helps a lot to understand how you would design then a phase 3 trials. So what you would see in the phase 2 trial would already indicate how you would set up your phase 3 trials. And you can, in the phase 2 trial, you can randomly assign patients to different treatment group. So they could be randomized, blinded, placebo, or active control trials. It really depends on what you want to see in your trials. And you can already see that from the phase 2 to the phase 3 trial, only 33% of the phase 2 trials make it to phase 3. So it's already eliminating a lot of potential drugs. The phase 3 trials are actually the ones which are, let's say, the most important, because they would determine if you would file for the with the regulatory agencies as the FDA or the EMA. It's usually large trial, more than thousands of participants, and it's to understand if the drug that you test is better than what's already available. So most of the time you would test your drug versus the existing standard of care. And you need, as an investigator, you would need to demonstrate that your medication is at least as safe and as effective as existing treatment option. Otherwise, you would think, what's the point to actually put a drug on the market which is less safe and less effective? And then you would use the randomization to actually understand how your drug works compared to a standard of care. These trials are usually quite long, and then this is where you can start to see the long-term side effects. And then this is afterwards, you would start then creating your label for the drug and for the condition. And with that, if your trial shows superiority, then you can actually start to get the approvals from the agencies. And if you success with your phase 3 trials, about 25 to 30 of them can be submitted to the agencies. So as you can see, it's really not so much. So you go from thousands of compounds to actually one compound, and when within this one compound, then you don't get that many trials, which allows you to actually file for some approval. So in the US, this process is called the new drug application. So it's to show that your trial and your drug is more effective and safer than current treatments. The FDA review the result of the trial and other information that you need to provide. And then they would decide if they approve or not the treatment for the condition that your drug was tested on. And then if it's approved, then this treatment often becomes a new standard of care, and then other drugs can be tested against it. So this is how the treatment landscape is usually changing. But the FDA can also ask you to run more studies in case they see that more information has to be provided. It's a pretty similar process in Europe, that's called a marketing authorization application. It's not so unusual that they would ask for actually 2 phase 3 trials before approving a drug. So it really depends in fact, on the disease, on the medication, on the unmet need, on the population that would benefit the drug. But it's not uncommon that they would ask for 2 phase 3 trials before granting a marketing authorization. And then once you have a marketing authorization, whether in the US or in Europe, you would go for its phase 4 trials, which mainly are observational studies. And it's really to gather information on what more you need to know. So you would need to understand the long-term safety and the long-term benefit and risk. And these trials are usually several years, and it's thousands of participants, because it's the first real life data that you get in the clinical process. So it's the physicians who are prescribing the drug to patients, and then you would collect the data directly from the patients and the doctors. And this is also where you start looking at quality of life, of cost effectiveness. So patients, they can get a drug used in a phase 4 trial without being in a study. So you don't need to be enrolled in a clinical study to get the drug. If you are in a clinical study, then you would be able to report what's going on, but if not, it would be voluntary reporting. So this is where you get the most information once the medication is approved. And there are still some misconceptions regarding some trials. So, do we use placebo? Not so much. We do not use placebo so much in interventional trials. Do all trials help to develop new treatment? Not necessarily, because it could be that the trials, it could be that you have to run a trial just to maybe expand a label, so expand an indication. And is it only drugs that have to go into clinical trials? And no, so it's also the medical device have to go via the same kind of process. So this is, misconception that we usually have about clinical trials. And so no compromise about quality. The biotech industry, the pharma industries are very highly regulated environments, and there's no compromise to be made with patient safety and drug efficacy. When you develop a drug, it has a huge cost, and there's a high pressure on the cost for the innovation cost a lot, the clinical trial cost a lot. And then we also know there's a lot of pressure which goes around the maybe alliances, business alliances that they make between some companies. And the time is also a big constraint. Time is money, as we know. And when you actually develop a drug, every day is a huge investment. But also, obviously, because you want your drug to be made available to patients. And as we know, also, investors are also putting a bit of pressure on that. There, we also have the pressure of the competitors. You want to be first on the market. And also, you have pressure with the health authority and also your partners if you have. And here is just a summary slide to show that, again, as said, you go from more than 10,000 compounds to actually have one drug approved after 10 years. So then you can understand that the drug development process is it's a heavy process and a costly process and not so easy process. So yeah, I guess you understand. So that's the end of the presentation for now.
Thank you very much, Samantha. That's really, really interesting and some good questions coming through. So I'm going to put you on the spot now. One of the things that wasn't heavily featured, and probably because it's not heavily featured in the process, is the understanding of the patient perspective and the patient need. At what stage is the clinical needs, at least, if not the patient's need, incorporated in that process, and particularly at the early stages?
So, so we know that ideally patients could be involved at any stage, and that's ideally. In the reality, it's not so much the case, unfortunately. Whatever the reasons are, I guess there's a lot of pressure put on the industry, and for the reason we mentioned, the quality, the time, the cost. So probably industry can do better. I'm not too sure that they have the flexibility right now to do more than what they are actually doing. I think they are really trying to do their best. So that would be my personal opinion. Then I think if you want to have more information, I would suggest that you join the next webinar that Jan Geisler will present on patient involvement in clinical development. He would probably be able to give another perspective.
Excellent, thank you. One of the other things that quite often appears, at least externally, when you're looking at the drug development process, is somewhat a lack of advanced planning. Externally, at least, it all seems very siloed within companies, and you mentioned it kind of offhand in your presentation about, and this is the point at which we start to think about cost effectiveness, rather than from a very early stage thinking about the clinical and the cost benefits of a treatment. Is that what it appears like from the inside?
Yeah, it does. It does. It's yeah, and I remember from my past experience that actually, when you work in a company, you even find out that the chemistry guys don't talk to the biology guys, or the chemistry guys do not talk to the medical guys in the company. So it is indeed very siloed. Yes. Yeah.
And then there were a number of slides where you showed kind of 10,000 drugs turning into one drug reaching the market at the end. What happens with the other 9,999 drugs lost along the way?
That's a good question. So it happens that sometimes they actually go back to one of the 99,999 compounds and they say, okay, maybe this one would actually fit our needs better. So but it's quite rare that they would do that. And otherwise, they may just test it for another disease or use the compound for another target. So it really depends.
And then one more question on the role of patient organisations, which is probably not surprising given the intended audience. How do we improve patient involvement in this? Maybe not in the short term, but in the long term, where are the opportunities to make sure that the drugs coming through truly meet the needs and things that are important to the patient community?
That's not an easy it's not an easy question. I think there's a big mindset that pharma has to change, and they have to understand that involving patients even in business decision is even cost saving or time saving, because they would already get the patient needs and the patient opinion much earlier on. But then we know also that it's a very highly regulated environment. They are also under a lot of constraints when it comes to the regulatory process, for example. There's not so much room for creativity when it comes to developing a drug. So probably the input that could be given is really about the unmet needs and the market opportunity, and maybe what kind of disease you would look at, or what kind of unmet need you would try to cover. Then when it comes to sales, unfortunately, I don't think patient could really be involved, or when it comes to the regulatory process, probably not so much as well. So it's not easy. It's really not easy. And it's, from what I've seen in my experience working in pharma, is not that industry does not want to involve patients. It's just most of the time that they cannot involve patients, unfortunately.
And certainly a difficult topic, and that's why we've got a whole webinar dedicated to it in our next session. And as you said, Jan Geisler is going to be here to talk specifically around the patient role in the drug development process. So that's all we've got time for in terms of questions. Thank you very much, everyone, for joining us. If you have any questions following this, then I'm sure you can contact Alan and Samantha specifically with any of those following that, or post them on the Facebook if you're watching here on the Facebook Live. And we look forward to seeing all of you at the next session with Jan next month. And I'm sure any of the questions you have may also be relevant there. So thank you very much again, Samantha, for the presentation. That was very helpful.
Thank you. Thanks.
Nice to see everyone. Take care and see you at the next webinar.
Bye-bye.